Hello children. So in our previous video we have talked about basics of respiration, and in the very previous video we had started with aerobic respiration. And basically aerobic respiration is the one which occurs in the presence of oxygen, and in the last video itself we had talked about we had taken the idea of the whole aerobic respiration, which includes glycolysis, link reaction, which is also known as acetylation, Krebs cycle, and electron transport chain. Also, we had talked about acetylation reaction in the previous video, wherein two molecules of pyruvic acid which were formed during glycolysis process now has converted into acetyl CoA. So today in this video we precisely are going to talk about Krebs cycle, which is also known as citric acid cycle or tricarboxylic acid cycle. So Krebs cycle is going to start with something known as acetyl CoA, which was formed during link reaction only, right? During acetyl reaction or the link reaction. So acetyl CoA, it is a 2-carbon compound, and this acetyl CoA now going to add up with another compound known as oxaloacetic acid. It is also abbreviated as OAA, oxaloacetic acid, and it is a 4-carbon compound. Now, when 4-carbon compound getting added up with the 2-carbon compound, a 6-carbon compound going to form, that is known as citric acid. So it is a 6-carbon compound. Now, during this reaction, what is happening basically, when acetyl CoA combines with oxaloacetic acid, it is happening by addition of a molecule of water. So I can say this step is also known as hydration. Hydration, right? And this step, it requires, of course, an enzyme, since citric acid is being formed here. So the enzyme that is involved here is citrate synthase. Citrate synthase, right? Now this citric acid that is being formed here, it is going to go some set of reaction, and the basic step here is isomerization. So citric acid is actually getting converted into its isomer known as isocitric acid. Isocitric acid, right? So the conversion of citric acid to isocitric acid is basically a 2-step process, wherein what is happening, first there will be removal of water, and then there will be addition of water. Okay, so I can say first there is dehydration is happening, and then there is hydration is happening, right? And both the process, it is occurring in the presence of same set of enzyme known as aconitase enzyme. And this aconitase enzyme, it requires a cofactor, that is iron. So iron will be there. Okay, so when basically citric acid is undergoing this dehydration process, there is an intermediate compound that is being formed, and it is known as, let me write it here only, cis aconitic acid. Okay, and ultimately this cis-aconitic acid, when undergo hydration process, get converted into isocitric acid, which is again a 6-carbon compound. right? Now this isocitric acid is again going to undergo some set of reaction. So let me write it here. So isocitric acid is first undergoing decarboxylation process. Decarboxylation process, wherein a molecule of carbon dioxide is being removed from this isocitric acid. So this step is, I can say, decarboxylation, right? And here also this isocitric acid is undergoing removal of hydrogen. So when hydrogen is being removed from isocitric acid, what is happening? It is going to get picked up with or from NAD plus, and it is going to get converted into NADH plus H plus. So since oxidation is also happening, that is removal of hydrogen, so this whole step can be termed as oxidative decarboxylation, right? And ultimately, since now here one carbon is lost and this was a 6 carbon compound, so whatever it is going to form here is going to be 5 carbon. So what is going to form here? Alpha keto, a compound named alpha keto glutaric acid. And since one carbon has been removed here, so we are left with 5 carbon compound. So this alpha ketoglutric acid is the only 5 carbon compound in the whole cycle. It is the only 5 carbon compound. And this step again is going to, or it has occurred again in the presence of enzyme. Since hydrogen is being removed, so the enzyme is dehydrogenase. And since isocitrate from the hydrogen is being removed from isocitric acid, so it is isocitrate dehydrogenase enzyme, and we are left with alpha ketoglutric acid, which is a 5 carbon compound. Now again, this 5 carbon compound, that is alpha ketoglutric acid, will undergo next set of reactions. So let me write it here. Alpha ketoglutric acid will again undergo first decarboxylation process. So here a CO2 is being removed, and also again here oxidation is going to occur. So there will be removal of hydrogen, which will be picked up again by NAD plus, and NADH plus H plus will be formed, right? Since again carbon dioxide is getting removed and oxidation is happening, this step is also known as oxidative decarboxylation. Oxidative decarboxylation. And simultaneously in this step itself, a coenzyme A is also being added up. So ultimately we are going to get another compound, that is succinyl coenzyme A, succinyl CoA, right? And since one carbon is being removed, we were at 5 carbon compound, one carbon is getting removed, so this succinyl CoA has to be a 4 carbon compound. And again the whole set of process is occurring in the presence of another enzyme, that is alpha keto glutarate dehydrogenase, right? Now this succinyl CoA is again going to undergo another set of reaction, that is removal of coenzyme A, plus also there will be elimination of one inorganic phosphate, and this inorganic phosphate is going to be picked up by GDP, that is guanosine diphosphate, and ultimately going to get converted into GTP. This GTP is guanosine triphosphate, which is equivalent to ATP only. It is equivalent to ATP only. Okay, so this is happening in the presence of another enzyme, that is succinyl thiokinase, and ultimately we are going to get a compound named succinic acid. So no decarboxylation has happened. So now we are not going to decrease the carbon number. So succinic acid again going to have same number of carbon, that is 4 carbon in it. Okay. Now this succinic acid is going to get converted into something known as fumaric acid. Fumaric acid. And this is happening because this succinic acid has gone oxidation process again. Oxidation process again. Right? So since oxidation is happening, there must be removal of hydrogen, and that hydrogen will be picked up by not NAD plus now, it is going to picked up by FAD plus. So here FAD plus is picking up this hydrogen and it is getting converted into FADH2. We write it again. Kind of messed up, right? Yeah. So FAD plus is going to pick up that hydrogen and going to get converted into FADH2. Right? So here this enzyme, this process requires another enzyme, that is succinic acid is getting converted into fumaric acid. So succinate, and since oxidation is happening, removal of hydrogen is happening, so succinate dehydrogenase enzyme. Okay. Now this fumaric acid is going to get converted into another compound that is known as malic acid. Again, what is happening during this step? Here, addition of water is happening. So I can say this step is hydration. And basically this step is where fumaric acid is getting converted into malic acid. It requires another enzyme as well. So fumaric acid is getting converted into malic acid, so the enzyme is fumarase enzyme. Right? Now this malic acid, again, see all the compounds that are being formed here now, succinic acid, all this fumaric acid and malic acid, these are 4 carbon compound only. Okay. Now this malic acid is going to get converted into ultimately oxaloacetic, where our cycle is going to complete. So malic acid is going to get converted again into oxaloacetic, in the presence of which enzyme, of course, during this step again what is happening, this is oxidation step again. So removal of water must be happening, and that hydrogen will be picked up by phos NAD plus, and it is going to get converted into NADH plus H plus. Right? And this step again required an enzyme that is known as, since malic acid is getting converted, so malate, and oxidation is happening, malate dehydrogenase. So cycle is completed here. We have started with oxaloacetic acid and now we are again at oxaloacetic acid. So again and again in the cell this cycle is running. It is getting started from oxaloacetic acid and ultimately getting ending up on oxaloacetic acid only. Okay. So this whole was your Krebs cycle. Which is also known as tricarboxylic acid cycle, so TCA, and it is also known as citric acid cycle, since the very first compound that is formed here is citric acid, right? So this whole cycle we have run for one molecule of acetyl CoA, children. But if you remember, during glycolysis from one molecule of glucose, we have formed two molecules of pyruvic acid, which again undergo acetylation reaction, and here we have got two molecules of acetyl CoA. So this cycle that we have just made was for one molecule of acetyl CoA. Okay, so in order to convert like two molecules of acetyl CoA, you need to run this whole cycle twice. The cycle will be running twice. Okay, so if I'm talking about one molecule of acetyl CoA, one molecule of acetyl CoA, then what would be the number of ATP production? So here during one cycle, only at one place the GTP is being formed, that is equivalent to ATP only, right? So I can say one molecule of ATP is being formed, right? And if I talk about just like that NADH, so here one NADH, right? Second NADH and third NADH, right? So I can just write it here, 1, 2 and 3, so that it will be clear to you. So in total 3 molecules of NADH has been formed. Just like that, if I talk about FADH2, it was formed only at one place here, so one molecule of FADH2, right? And if I talk about carbon dioxide, sometimes they can ask even about carbon dioxide. So for carbon dioxide it is 1 and here 2. So 2 step was decarboxylation. So 2 molecules of CO2. And this I talked about with respect to single cycle, one cycle of Krebs cycle. If they ask you for 2 cycle or 3 cycle, you just have to multiply this whole things with 2, 3 or whatever has been asked. Okay. So this was about your Krebs cycle, and we are left with only one thing of aerobic respiration, that is electron transport chain, which I'm going to talk about in the very next video. So stay tuned and I'll see you next time. Bye bye.